I'm here to talk to you today about something that medicinal genomics has developed, and I think it's going to revolutionize testing using qPCR methods. So I've been working at Medicinal Genomics as an R&D scientist for the past 5 years, and over those past 5 years we've developed a lot of tools to help the cannabis community, particularly in the genetics of cannabis, as Kevin talked earlier about Jamaican lion, and in microbial testing. So microbial testing, while it's not the most glamorous, it is very important. Every state that is legal to sell cannabis has potency, heavy metal testing, and microbial testing. And like I said, it's not the most glamorous, but it is the most important because the only confirmed deaths in cannabis have come from microbial contamination, Aspergillus in particular. Although with the recent controversy with vape pens, that might be up for debate, and I think there's some talks on that maybe tomorrow. So, as we've just heard, cannabis is used as a medicine, and there are immunocompromised patients out there, some that don't even realize that they're immunocompromised. So it's really important to make sure that the cannabis that they're receiving is free of microbial contamination. So it's imperative that the testing labs have the ability to accurately detect for harmful microbial contamination on cannabis. And there's generally two methods that are out there for microbial testing: molecular methods, and we at Medicinal Genomics have qPCR testing, and culturing methods, which is your traditional, does it grow on a plate? And they both have, as you can see, there's qPCR methods have more pros than culturing methods in that it's faster, it's more accurate, you can specifically target the contaminant that you're looking for, and it's scalable. Whereas culture methods are slower, they're more inaccurate, they're not specific, and it's harder to scale. But the one thing that qPCR lacks that culture methods has is that, can I go back without messing up? Is that if the DNA is present in a qPCR assay, it's going to be detected. It doesn't matter if it's live or if it's dead. So there's two types of testing in cannabis microbial testing: qualitative or presence-absence testing. And this requires that no amount of the target organism be present in 1 gram of sample. So these are your 4 Aspergillus species: Aspergillus flavus, Fumigatus nigrotarius, Salmonella, Stach, and E. coli. And then you have qualitative, or the total testing. And this allows threshold limits of there to be microbial contamination. So for example, total yeast and mold, most states have a 10,000 CFU limit. Total aerobic bacteria, 100,000 CFU. So like I said before, qPCR detects all DNA, whether it be live or dead. And this can be a concern in the cannabis industry because there are some cultivators out there that want to make sure that their cannabis is free of this microbial contamination so that it will pass. And so they're doing techniques like irradiating. They're killing the microbes. But killing the microbes still leaves behind their DNA, and qPCR methods will pick up that dead DNA. But we've developed a method to solve this problem, and it's called Reefer. So like I said before, the pro of qPCR testing is that there are primers and probes specific to amplify the DNA that you're targeting. However, it cannot distinguish between live or dead DNA. And the solution that we came up with is eliminate the contaminating dead DNA, again, by this method called Reefer. So I'm just going to go over, let's see, how this, if I point to there, will it show? So it's a simple, upfront, 10-minute incubation at 37 degrees Celsius. What you do is you add in your Reefer enzyme along with a buffer into your sample. This all happens before DNA extraction and lysis. The Reefer enzyme comes in and it chews up any of your free-floating or dead DNA. After 10 minutes at 37C, you add in the MGC lysis buffer. that lysis buffer deactivates the enzyme. The lysis buffer, while deactivating the Grim-Reefer enzyme, also opens up the cells of your live organism. Thus, now you have only your live organism's DNA present. And then what we've added is we've added a positive control into this mix. And this positive control is a DNA template that you add in to ensure that your enzyme is in fact has been deactivated. And that will later down the line be tested in your qPCR reaction. So after the positive control, after you add in your lysis buffer, you add in your positive control, and then you proceed with the MGC DNA extraction procedure. And that positive control is in there because if you have your original method versus your Grim-Reefer method, you expect that that positive control DNA is not going to change. It should be exactly the same CT value as the original method versus the Grim-Reefer method. So to test this theory, we took a live E. coli culture. And this was an E. coli culture that had only been growing for 4 hours to ensure that the majority of the culture was in fact live. And we split that into, we put that in the presence of hemp and put it into 12 different tubes. 6 of the tubes, I think that was the wrong button. 6 of the tubes were treated with the original method. 6 of the tubes were treated with the Grim-Reefer method. And increasing copies of Salmonella DNA were spiked into each tube. They were treated 10 minutes, 37 degrees, and then after the 10 minutes, the MGC lysis buffer was spiked in to stop the enzyme, and the positive control was added, and DNA extraction happened. So 12 samples, it was run in triplicate, so a total of 36 qPCR reactions were run. So these are the results for E. coli. And so the hypothesis is that due to the presence of E. coli, the E. coli results for the Grim-Reefer method may be lower than the original method, but since E. coli was freshly cultured, E. coli results will be more similar between the methods than the internal control results. And again, like I said, this was only a 4-hour E. coli culture. And as you can see, there's no statistical significant difference between the E. coli results and the Grim-Reefer results. So on your x-axis, you have the increasing amount of Salmonella DNA, and on your y-axis is the cycle of qPCR, cycles of qPCR, goes to 40. And the results are consistent with the hypothesis that the Grim-Reefer method does not interfere with DNA from live cells. And both methods demonstrated robustness in that there was no impact from the increasing amount of Salmonella DNA. And so these are the Salmonella results. And the hypothesis here is that free Salmonella DNA will not be detected for the Grim-Reefer method until the amount of free DNA exhausts the enzyme. And as you can see, the Grim-Reefer method consistently deactivated the Salmonella until you got to the 4.02 million copies. And you see that purple bar there is the Salmonella signal. In qPCR results, the higher the signal value, the less DNA is there. And so the results from the Grim-Reefer method are exclusive of free DNA as long as DNA is less than 4.2 million copies. And just to give you an idea, a 10-fold dilution is represented by a 3.3 CT shift. So for the 6.03 million copies, there was an approximate 12.93 CT shift, which is about a 7,500-fold reduction in free DNA. So now here's the Grim-Reefer positive control. And as I said earlier, that the hope here is that the enzyme is deactivated with the addition of the MGC lysis buffer, and therefore you should not see any difference in your Grim-Reefer positive control between the two methods, and that's exactly what you see here. The null hypothesis cannot be rejected in the 95% confidence interval since the p-value was 0.161. So there is no statistically significant difference between the Grim-Reefer control and the original method. And this result is consistent with the hypothesis that Grim Reefer enzyme is no longer digesting free DNA after incubation and after a lysis buffer is added. And both methods demonstrate robustness as there's no impact from the increasing amount of Salmonella DNA. And so another feature that the MGC qPCR assays have is we have built in an internal control, and that's targeting the cannabis DNA. And so due to the inherent presence of free DNA in the plant material, you would expect that a dried sample is going to have some dead DNA. So you would expect to see the internal control shift between the original method and the Grim Reefer method. And that's exactly what we see here. The null hypothesis, the Grim Reefer and original method internal control results are the same, can be rejected at the 99% confidence interval since the resultant p-value is 2.45 times 10 to the negative 25th. So there is a statistically significant less hemp DNA detected in the Grim Reefer method versus the original method. And the results are consistent with the hypothesis. So these are some qPCR curves. This is utilizing 4 different channels in the instrument, the FAM, HEX, Sci5, and ROX channel. And let's see if I can get this to work. So the orange signal that you see here is the Salmonella signal. And this is at 402,000 copies. And as you can see with the Grim Reefer method, that orange signal goes away. And then when you get down here at the 8 million copy levels, you see the Salmonella signal very strong here in the orange, and it decreases after the Grim Reefer method. So this is an approximate, I believe it's a 12 CT shift here. So here's some conclusion summaries. The Grim Reefer method is not metabolizing DNA in viable cells. It's not metabolizing DNA after deactivation. The Grim Reefer method is capable of metabolizing free DNA up to 4.02 million copies. Both the original and Grim Reefer methods demonstrate a high level of precision. Both methods demonstrate robustness for all analytes observed, as there was no impact for the increasing amount of Salmonella DNA. And the original method results are proportional to the amount of free DNA plus DNA in viable cells. And I apologize for the numbering being wrong there. So now that we had shown that this worked in the lab, we wanted to see what would happen in real cannabis samples, because we're not a cannabis testing lab, so we don't have access to actual samples. So we asked 4 independent labs if they would be interested in beta testing this. And they said yes. And so now I'm just going to take us, so there's a lot of results. I'm just going to go over a snapshot. I kind of picked some from the total aerobic assay, and I just wanted to point some things out here. So lab 1, what you can see here is that there is a shift in the TAC signal. It goes from 25.2 to 27.95. There is also a large shift in the SCCG signal. That's that internal cannabis control. You see it go from a 26.21 all the way to a 32.36. And the Grim Reefer positive controls are very similar for the 2, and that's what you would expect. Lab 2, with the original method, there was a detection at 25.87, but with the Grim Reefer method, none was detected. And this indicates that most of the DNA that was present on this sample was in fact dead. And you can see that there was a large HEX SCCG signal shift here. Yet the Grim Reefer method positive control remains the same. So I don't know what this sample could have experienced, but with the large shift in SCCG signal, this could have been a sample that had been heavily irradiated. Lab 3, you can see that the TAC signal isn't really changing all that much. So this is indicating that most of the organisms present on this sample are in fact alive, as you're not seeing that CT shift. And there's about a 3 CT shift in the SCCG signal, and again, the positive control in the Sci5 is similar. And finally, lab 4, you see a very large shift, again, in that TAC signal 26 to 37. The hex signal, there's also a shift, and again, the internal control signal, the positive control is similar. So there are other free DNA removal products out there. One is PMA, and PMA is a high affinity photoreactive DNA intercalating dye, and what that basically means is that the dye goes into the double-stranded DNA, and it becomes activated when it's exposed to light. He's in my way. And this results in a permanent DNA modification, which renders the DNA insoluble and therefore can't be extracted through your standard DNA extraction methods. The dye is cell membrane impermeable, so that means that it can't get into the cells of live DNA. There's also other enzymatic treatments out there, and they require a 15 to 30-minute digestion. But they also require a 15-minute inactivation step at 95 to 100 degrees C. And that inactivation step at such a high temperature is going to cause the live cells to start to lyse. And then you're going to have a competition happening between those cells that are starting to be lysed and the enzyme being deactivated. So these both have their faults. PMA, it requires an incubation in the dark followed by exposure to visible light, either through an LED device or a halogen lamp. PMA does not work equally across all microorganisms, and different microorganisms may require different concentrations of PMA and different exposure to light. And PMA is toxic, which creates a biohazard waste stream. As for the other enzymatic treatments, the product literature that they claim has an average signal reduction of 2 logs, or about 6 CTs, which isn't as good as what I had just showed you. And again, what I had just stated before, the heating the sample to 95 for enzyme inactivation will result in the lysis of live organisms. So some final thoughts. Grim Reefer offers a simple 10-minute 37C upfront incubation that is non-toxic and easily deactivated without causing harm to viable organisms. It results in a 3 to 4 log or 12 to 13 CT reduction signal from free DNA. The method includes an added positive control to monitor deactivation of the enzyme. It can be easily added to the current MGC DNA extraction protocol and provide qPCR results that are void of dead DNA. And this can be used to guide sterilization protocols at grows. I'd like to acknowledge our collaborators that helped us work on this project, and of course, the team at Medicinal Genomics. Anyone have any questions? What I do?
Thanks for the talk. Actually, I had a quick question, which might be obvious to folks that are in the industry here, but I was just wondering if it sounded as if the microorganisms are dead, then it's not a concern anymore. But if you had like a massive mold infestation on the product and then killed it with radioactivity, would there still be toxins and other things associated with it? I'm just curious why the distinction between living and dead.
So it depends. If you had a major Aspergillus contamination, which produces microtoxins, then yes, I think it would be a concern. I'm not sure. Every state has different rules in their regulations as whether if they fail, can they go back and be remediated and tested again. I was under the impression that in California, it doesn't matter if it's live or dead, but I could be wrong. So I think it depends. But there are other regulations that do test for mycotoxin testing. So if it was dead and left something behind, I would think it would come up positive there.
Yeah, this was a really interesting talk. I not working in this field, I was a little confused at first when you described this as dead DNA. Isn't it that your test is detecting any free DNA attached to the cells? And if for some reason there was cell lysis that occurred during the preparation of the extract, would that skew your numbers?
Yeah, it could. And we call it, so a lot of it's basically viable versus non-viable. So in culture methods, you're only detecting living organisms, cells that are able to create colonies on your plate. Where in the qPCR, it doesn't distinguish between the two. Yeah.
It's agnostic.
Yeah.
Okay, thank you.
Yep. Great. Thanks, guys.